Okay. 2 Brunos now.
Yeah.
Oh, 2 Brunos?
Yeah, you should.
That was supposed to be the meeting thing. Oh, but it's okay. 2 Brunos now. I'm cloned. Oh, I'm doing 2 jobs anyway, so I'm cloned. Okay, this is fine. I'm using. Have you guys have used
2 Brunos now.
Yeah.
the OBS? Oh, 2 Brunos. OBS? Oh, really? On YouTube. Now there's a sound. I'm gonna restart again. I'm sorry. That's not the first time I do this. Okay, guys.
It's no sound.
Yeah, I know. They warned me. I was giving a whole speech here with no sound. Anyway, so thank you again, everyone, for coming for one more lecture. We are going to start this new roundtable. It's wait, there's a sound here behind me. I'm a little confused. So we're gonna start here this next roundtable. I'm very happy to have here Gu and Marc in this talk, this roundtable. I'm glad that everyone is here right now too. I hope you guys enjoy. Just to remember, 1 second. I'm kind of I'm having a delay here. I'm confused. Anyway, so to be less formal, since I'm doing a lot of stuff wrong here, but anyway, First of all, it's going to be Marc who's going to talk, give his talk in this talk, this roundtable about neurodevelopment in zebrafish. Then it's going to be me, and then it's going to be Bruno. I'm going to ask everyone to write your questions on YouTube chat. Feel free to write in Portuguese. It's no problem at all. I'm going to ask them the questions. You guys are going to be fine. Marc, thank you very much. I would say like the floor is yours, but that doesn't work here. So the screen is yours.
Okay, can you see it, Bruno?
Yeah, perfect.
Okay, so good afternoon, everybody. First, I'd like to thank Bruno, Beatrice, and the other organizers of this symposium for the kind invitation to talk about our work here. Today, I'll tell you a little bit about some recent work we did in trying to kill dopaminergic neurons in zebrafish using several approaches and see if the fish is able to respond with an increased production of these neurons. So as you know, the fact that zebrafish is able to regenerate lots of cell types, lots of structures after injury or disease, has made this fish an interesting model to study neurodegenerative disorders, including Parkinson's disease. Another feature of the fish that makes it particularly useful to look at what's happening in the adult brain is the remarkable ability of the zebrafish to generate new neurons throughout its life. And on the slide, you can compare the neurogenic potential of the mouse on the left side and of the fish, and the fish has 16 or so neurogenic zones and a large number of adult stem cell niches in the adult. We are going to focus primarily on the dopaminergic system. The brain of the fish has some similarities to that of mammals, but of course there are also notable differences. But a lot of labs have examined the different clusters and centers of dopaminergic neurons in the brain. A lot has been done during embryonic, larval, and juvenile stages, but also it has been studied in the adult. So a number of years ago, our lab started by producing 3 different lines of transgenic fish that we thought would help us analyze dopaminergic neurons. And in order to do this, what we did is we used the locus for the dopamine transporter, and we used a large region of this locus, more than 25 kilobases of DNA. And what we did is introduce in frame with the first exon of the dopamine transporter a number of fluorescent protein or other proteins. And that's what's shown on this slide. We did it for with EGFP, and that allows us to label dopaminergic neurons. We did it with a version of mCherry that is also fused to the mitochondrial localizing signal of the TOM20 gene. And that allowed us, because it is driven by the regulatory element of the dopamine transporters, to target specifically the mitochondria, or label specifically the mitochondria of the dopaminergic neurons. And finally, as you will see in the last part of the talk today, we've made a fusion protein between nitroreductase and the cyan fluorescent protein in order to do the chemogenetic ablation of the dopaminergic neurons. So, as I mentioned, the gene we took advantage of to target our transgene is the dopamine transporter, which is responsible for the reuptake of dopamine into the nerve terminals. And if you look at expression of this gene in zebrafish embryos, for instance, if we look at here, we look at the expression of the dopamine transporter in the ventral diencephalon, and we see this typical horseshoe shape that is characteristic of the dopamine cells at that level of the brain at that stage of development. So, with these tools in hand, we decided to look into ways to ablate dopaminergic neurons and look at what happens in terms of their regeneration. And I must admit, we are one of the many labs who have used such approaches, especially using neurotoxins to kill dopaminergic neurons. But when you kill dopaminergic neurons, by reviewing the literature, we see that there were a number of issues. There was some variability in the outcome and the physiological and behavioral consequences of ablation. And when it comes to the brain, as you will see in part of my presentation, but as we could also see the uniformity of the effect of these treatments to ablate dopaminergic neurons could be an issue. And finally, the amount of reductions that you are obtaining can also be a problem that you need to overcome for a number of reasons. First, as we know from Parkinsonian patients, you need to achieve a very large reduction in the number of dopaminergic neurons before you can observe behavioral consequences. And the second problem with the percentage of reductions is that when you're working with zebrafish, either at the juvenile stage or in the adult, you're trying to kill neurons in a system that is producing these neurons at a fairly high pace, especially during development. So obtaining large reductions was also an issue. So I'm going to divide this talk into three parts, three recent studies we have done. First, I'll tell you about a survey of neurotoxins that Michael Kalin, a student in the lab, has been doing. Then I'll tell you about our efforts to use intracerebroventricular injections into the adult brain to ablate dopaminergic neurons. And finally, I will resume what we've done summarize what we've done with chemogenetic ablations. So, neurotoxins. As I was mentioning, there's a very large number of studies of people who have used neurotoxins to ablate dopaminergic neurons. And by going over these studies, we were trying to first figure out what would be, if we were to use a neurotoxin, what would be the best one to use, and try to see if by testing several neurotoxins at once, we could get some sort of uniformity in the effect, or trying to make some sense as to the relative potency of these neurotoxins. So that's what Michael did, and he tested using a similar schedule of administration. MPTP, the MPP plus, which is the active form of MPTP after it's been metabolized, paraquat, 6-hydroxydopamine, and rotenone. And this work that Michael did was done in juveniles, in embryos. And for some of the slides that I'm going to show next, we've divided this ventral diencephalic presence of dopaminergic neurons into clusters that were kind of defined by colleagues, others. And here on this slide, we see using a similar route of administration in the water of the fish, using doses that were based on published literature and using the same time of administration and the same time at which we measure. Well, when we compare MPTP, MPP+, 6-hydroxydopamine, paraquat, and rotenone, we found that in our hands at least, MPTP, MPP+, and rotenone were those that gave us the strongest reduction in numbers of dopaminergic neurons. If you compile what happens in clusters 8, 12, and 13 that you saw on the previous slide. Then what we looked at are swimming parameters in these fish, and again, MPTP was and rotenone were the two that gave us the strongest effect, both in terms of swimming distance and of the swimming velocity. So what I didn't have time to show you is that we also took into considerations the non-specific effects of these drugs in terms of the deformities that we could see, the mortality, and so on and so forth. And in the end, we decided that we would stick with MPTP. But then Michael switched to the adult brain, because the previous study that I just showed you was done in embryos. And he decided to do cerebroventricular and micro-injections of MPTP that were injected at the level of the anterior optic tectum. And when Michael examined what's happening in terms of dopaminergic neuron numbers after 1, 2, 3, or 4 injections, he said they could obtain noticeable decreases in the olfactory bulbs and kind of less profound decreases in the telencephalon of these fish, as shown here for the olfactory bulbs, the telencephalon, and here is another area that we've been paying particular attention, the periventricular pretectal nucleus. Using the fish that express M-cherry in mitochondria, we also observe some parameters that are indicative of the morphology and the fusion-fission dynamics of mitochondria in these fish. And basically, what we were looking at is whether the administration of MPTP could lead to an increase or of fragmented mitochondria, or as a reverse, a decrease in the number of the fused mitochondria. So this panel on the left shows you examples of the various categories of mitochondria. And following the intracerebroventricular administration of MPTP, we saw that the number of fused mitochondria decreases dramatically in the fish that were treated with the neurotoxin, whereas the number of fragmented mitochondria increased. And finally, Michael examined swimming behavior. Again, the duration of inactivity after the number of injections of the MPTP, and then leaving the fish to recover for a few days to see what happens. The average velocity of the fish and the total distance that was traveled by these fish. And after 3 and 4 injections, he saw these decreases in velocity and distance traveled. In comparison, of course, that is accompanied by, or that is accompanied, yes, by an increase in the inactive duration. And here are just the scans of the swimming behavior of the fish after the 4th injection, the vehicle controlled through the MPTP. But then after a period of recovery, you see that these decreases in distance or velocity are the fish has recovered from the impact of MPTP treatment. And finally, what Michael did is look at the olfactory response using the following apparatus. So in this apparatus, the fish is swimming into a tank that contains two compartments. And what we do is we inject or we dissolve into one of the 2 compartments, let's say this one, a nociceptive stimulus to the fish. It's the compound called cadaverine. And a normal fish, after you've injected or you've introduced cadaverine in this compartment, will have a preference for the other compartment or to the neutral zone. And we wanted to know if after ablation of the dopaminergic neurons using the intracerebroventricular injection of MPTP, this olfactory response would be affected. And indeed, it is the case. So in a control animal where we did not inject the MPTP, but only the vehicle, the fish spends a relatively small amount of time in the arm of the apparatus where we put the cadaver in, as shown here on the top slide. However, if the fish has been treated with MPTP, it swims about equal time in both compartments, presumably because it is no longer able to respond to the olfactory nociceptive stimulus. However, if you leave the fish to recover for a while, then the amount of time spent in the compartment where it's not supposed to be normally going has reduced to levels that are comparable to that of fish that only received the vehicle. So finally, in the couple studies that were done by former PhD student Rafael Godoy, who's actually from Brazil originally, Rafael used the CFP and TR fish to chemogenetically ablate dopaminergic neurons, and he first did it in a paper we published 6 years ago in embryos and larvae, and then looked at what would happen if the chemogenetic ablation in adults. So first of all, just a reminder, we've introduced into the DAT locus, the dopamine transporter, a fusion protein that contains CFP, cyan-fluorescent protein, and the nitroreductase, and that allows us to it facilitates screening of the fish, but at the same time, it also allows us to label the cells that express the transgene. And I'm skipping a little bit of it, but what Rafael was able to show is that if he administered the metronidazole, which is the prodrug that the nitroreductase will transform into a cytotoxic compound, well, if he starts to administer metronidazole to start the chemogenetic ablation at 1 day post-fertilization, and he continues this treatment until 4 days post-fertilization, and then looks at the number of dopaminergic neurons at 5 dpf, he obtained reductions in many parts of the brain, including the pretectum, the olfactory bulbs, the ventral diencephalon, the caudal hypothalamus. We also, if we looked 1 day after the stop of metronidazole administration, so 1 day after the end of the treatment to chemogenetically ablate the cells, we observed a transient decrease in dopamine levels. But as you can see, if you look again 1 day or 2 days later, the levels of dopamine have gone back pretty much to normal. Finally, Rafael looked at a large number of swimming parameters in these fish, and there was a decrease in total distance traveled when you look 1 day after the end of treatment at 5 dpf, and this gradually, or in fact rapidly, goes back to normal shortly afterwards. And these are other parameters that Rafael examined. So that's I'm skipping a bit here, but that's in a nutshell the study that Rafael published for the chemogenetic ablation at early stages, in embryonic stages. But we saw that the CFP and TR transcript is still expressed in the transgene is still expressed in the various parts of the dopaminergic system in the adult zebrafish brain, and therefore we attempted to do chemogenetic ablation in the adults. And for this, what we've done is to administer the metronidazole directly into the fish water, just like what we had done for embryos and larvae. But I'll try to remember to comment this later on. So here's the schedule of administration. The metronidazole was administered at 10 millimolar for a day. And then we examined the fish at several days after treatment, and we left some fish to go for 6.5 weeks to look at whether there would be some sort of recovery. And when you look at dopaminergic neuron numbers in 3 parts of the olfactory bulbs, we saw good size reduction in the number of cells after treatment. However, what we looked at then was to look at a few days after, several weeks after, in fish in which we had administered BRDU to look at cell proliferation. And as I mentioned at the beginning of the talk, fish continue to make new cells, including new neurons, throughout their lives in the brain. And when you look at the number of BRDU positive cells at 45 days, we saw no differences between the fish that received DMSO, these are the control fish, and those that received the metronidazole. However, if you look at the cells that were CFP positive, that is, they are dopaminergic neurons because they express the dopamine transporter and therefore the CFP-NTR transgene. And when you look at 45 days, we saw that there was a specific increase in the number of these cells. Okay, so the conclusion from that slide basically is that although, because we're looking at all cell types, the number of new cells probably didn't increase that much in those fish following chemogenetic ablation of specifically the dopaminergic neurons, but the number of dopaminergic cells in these fish was increased in those fish that underwent the chemogenetic ablation. Raphael did the olfactory test again, and what happened is that indeed, if you shortly after treatment, what we see is that, let's say, if we look at 7 days, there was an increase in time spent by the fish that were chemogenetically ablated, the time that they spent in the arm where the nociceptive stimulus has been added, a place where the fish would not normally go, okay? But this affected or altered or impaired olfactory response that was observed 7 days after the metronidazole treatment, while the olfactory response was, if you looked 6.5 weeks later, then the olfactory response was pretty much back to normal. But unfortunately, in this experiment, when we looked at various time points after the stop of the metronidazole administration, we could not see much impact on locomotor behavior. And what we think might have happened, it might have something to do with the way the metronidazole is administered directly into the fish water. And now we're trying to tweak the method to see if by administering the metronidazole by intracerebroventricular injection directly into specific areas of the fish brain, whether we could get more profound effects in terms of chemogenetic ablation in the adult zebrafish brain. So in summary, what I've told you today is that we are able to cause reductions in dopaminergic neuron numbers, and these are at least high enough to translate into behavioral deficit, okay? I mentioned to you that in the chemogenetic ablation in adults, we didn't see any locomotor effects, and perhaps it is just because the reductions in dopaminergic neuron numbers that we were able to achieve in the telencephalon was just not high enough to do that. So reminder, in Parkinsonian patients, you need to get fairly large reductions in number before you start seeing the signs of the disease. Okay, and we have, as I've shown you in the work that was done in the adult, we are having evidence for specific regeneration in the brain of the adult fish. And as I said many times during the talk, the uniformity of the effects that we can obtain either with neurotoxins or with chemogenetic ablation, probably something that requires that we develop improvements to the various methods. So I'd just like to finish by thanking the people who did the work. So Michael Kalin did the neurotoxin work. Raphael, who's not, I don't think, on the picture, did the work on chemogenetic ablation, and in that he was also assisted by a former PhD student, Kang Hua. And these are other people who are involved in several aspects of the dopamine Parkinson's / project in the lab, including Sandra Noble and Yan Wei Shi, who produced the transgenic lines you've seen in this talk. And this work was funded by NSERC, the Canadian Institutes of Health Research, and by the Ottawa chapter of the Parkinson's Society. And I thank you for your attention. And I'm not sure if we're doing questions right now or if we're doing it later, but I'll gladly entertain them.
Thank you, Marc. Yeah, I think we're gonna do all the questions later, because then we have, it's easy to link all the talks. So we're gonna do all together later. Is that okay for you?
Absolutely.
Okay, perfect. Thank you very much for your presentation. And now it's gonna be my turn. Guys, I just looked to the other side because it's the screen of where I put everything on YouTube, okay? So I'm just gonna first, here it's my turn, and then it's gonna be Professor Su. So first I'm gonna organize myself here, present a window, which is this one. Let me see if it's working so far. Yeah. Then I'm gonna go full screen. Done. And then I'm gonna do it this way. Okay, perfect. I think I'm fine there. Well, so now it's my turn. What I'm gonna talk to you guys is I'm gonna talk about the role of dopamine and dopamine signaling in the regulation of the development of the brain and the behavior. As some of you know, I'm Professor Bruno, and I work in the laboratory Neurodev at Universidade Federal de Minas Gerais, Federal University of Minas Gerais, right? It's not easy to translate that in Portuguese in my mind right away. Okay, it's working there. So to show what we've been doing here in the lab, first of all, I would like to introduce or to say some stuff about like the spectrum of behavior and the spectrum of cognition, and also the spectrum of neuropsychiatric disorders. So when we think about neuropsychiatric disorders, we don't have packages. People are not packed in different disorders. It's very difficult. It's getting more and more difficult to understand what is behind the neuropsychiatric disorders. So when we think about schizophrenia, bipolar disorder, social phobia, I can't read that, pure mania, all this stuff together, they all have a little bit of overlapping in a lot of the symptoms, and overlapping a lot of alterations in the brain, and also overlapping in a lot of the genes that are related to a lot of neuropsychiatric disorders. But of course, we have to, as a scientist, we have to find one little piece of the puzzle, and to think what kind of this little piece I'm going to study. Of course, we always wanted to study the dynamics, the systems, but it's very difficult to study altogether, right? It's a whole career to study just one little piece. So we just want to study one of the little piece in the puzzle of neuropsychiatric disorders, or even cognition, development of cognition. And the one piece that is here is dopamine. So dopamine, that's the little piece that we want to study. And why? Why did we choose dopaminergic system to understand development of the brain? First of all, we have a lot of evidence, a lot of different kind of evidence that support the role of dopamine or the dysfunction of the dopaminergic system in specific disorders. So we have pharmacological evidence in schizophrenia, bipolar disorder, ADHD, and a lot of genetic evidence for the same disorder as well. And recently, I wouldn't say recently, but in the last 30 years, we also are understanding more of the dopaminergic intracellular signaling alterations in a lot of different disorders as well. So when we think here, like here would be like the postsynaptic dopaminergic signaling. So we have here the D1 and D2 receptors. D2 receptor is specific, isolated. And as we know, they both regulate the activity of adenylyl cyclic, which produce cyclic AMP, PKA, and DARP32. So DARP32 is phosphorylated by PKA. So when we think of this, the dopaminergic intracellular signaling, and we checked in schizophrenic or bipolar or different disorders, so the proteins related to this signaling, we can see there's alteration, a decrease of DARP32 in the prefrontal cortex of schizophrenics, for example. AKT, there's a decrease as well. NCS1, there's an increase. Calcium, there's an increase. So we see all these alterations of dopaminergic intracellular signaling proteins in a lot of psychiatric disorders in brains of psychiatric disorder patients. So first of all, we started to studying some patients' lymphocytes to see if we could use some of them, like NCS1, DARP32, as biomarkers. We could see some of them. There's a decrease of NCS1 and CD4 positive cells in bipolar disorder patients and schizophrenic patients. We saw also an increase of DARP32 in bipolar disorder patients, but not in schizophrenic. So we saw that, well, that was interesting. There's a lot of studies to do to see if this could be a biomarkers or not. But to use these cells as a good model to understand the signaling pathways, it doesn't work, since we have exactly the opposite. In schizophrenic, we have an increase of NCS1 and decrease of DARP32. So we're not seeing that. It's not modeling what we want to see as thinking of neuronal function. Also, we did like a cell using PC12 cells. We increased the levels of NCS1 in these cells. And in these cells, we saw a decrease of DARP32. So we can see here the cells that increased the number of the levels of NCS1. We also saw a decrease of DARP32. Perfect. At that time, I thought, like, it's a beautiful data. Let's now treat it with antipsychotics and see what happens with this. But then when we treated with antipsychotics, we saw there was no alteration, not even in vivo, not in vitro using these cells. So for that, at that time, I thought, like, okay, something's happening. Like, I can see that when I increased NCS1, that activates, it increased the levels of activations of the D2 receptor, for example. We see a decrease of DARP32, but it's not rescuing or not with antipsychotic treatments. So with that, I got a little bit frustrated, as usual, as any scientist, and with frustration, we get new ideas. And in one data from another study that we were studying, another study that we were performing as well, we checked the effects of methylphenidate in the brain, treating young rats and adult rats with methylphenidate for 28 days. And what we saw is that when the rats are adult, they have an increase of NCS1 with the treatment with methylphenidate, but not the young rats. And when we see DARP32 levels, we see a decrease of DARP32 levels with the treatment in young rats, but not in adult rats. And that was the spark in the group when we thought, like, wait, we're seeing completely different reaction effects in different ages. So why not study like DARP32 and dopaminergic signaling in the developing brain? And then we had the following idea. Like, with all this data, we thought, like, okay, let's start studying the role of dopamine in different stages of development. Since when we think of the brain development, in the beginning of the brain development, we have a lot of neuronal proliferation, neuronal migrations, and then we have a very interesting window with myelination, synaptogenesis, apoptosis. So we have all this thing happening, but of course they overlap, but there's like an increase, like a more synaptogenesis in one part of the window of development, and the other part is going to be more neuronal migration. So we thought, like, well, dopaminergic signaling proteins, they're expressed in humans, for example, from 7th week. In mice and rats, in the 14th day of development in the embryo, and zebrafish 18 hours post-fertilization. So we thought, like, maybe dopaminergic signaling it has different functions in different developmental windows of the brain. So because of that, and using this hypothesis, we started to perform a lot of studies. In some of the studies, we used mice. And using mice, when we treated the mice, females and males, with L-DOPA to increase the dopaminergic levels, the dopamine levels between the first day of after birth and the 5th day, we can see that when they become prepubert, that was a tough word, at 4 weeks, we can see that females, they have an increase of locomotor activity or exploratory activity. And when we treat the adult, when we check the male mice behavior, we can see there's alteration in sucrose preference. So we can see like there was some alterations of imbalance, the dopamine signaling in the beginning, in the first 5 days of life of mice. We can see an imbalance in when the mice are 4 weeks old. And then when we check the brain, we could see that if we injected SKF3A393, so D1 agonist, in the first 5 days, we can see there's a decrease of arborization of the neurons in the hippocampus. And we can see that there's a slight decrease in L-DOPA treatment. There's a decrease with SKF, but it doesn't happen with Kipro. So we started to understand now which receptors are involved in that. But even though changing the arborization of these neurons, of the cells, we don't see alteration in the volume of hippocampus. So what is happening? Well, that's a question that I can't answer you guys right now. But that's something that we are studying. We're still checking that. And when we treated it for 14 days, so the first 14 days of life, and we check the 4 weeks, we can see there's a little bit more alteration. It increased the alterations of behavior of female mice and male mice. And interesting, when we check the 8 weeks, which means like when they were adults, so we saw that alteration when they were young, and now we're checking when they are adults, so 8 weeks, we could see that females, they have even an increase of alterations, an increase of behavioral alterations when they got older. So when they became adult. So after the pruning, the neuronal pruning, they changed even more the behavior. So you can see here that when we imbalance the dopamine in the first 5 days, and we check the behavior when they were 8 weeks, we could see an increase of locomotor activity, an increase of anxiety-like behavior, and an increase of sucrose preference. But when we checked the males, what we saw, it was a resilience, a little bit of, they didn't react that much. They had like anxiolytic-like behavior. I wouldn't say that, but they reacted less to the test of the forced swimming. In fact, it would be less depressive reaction, but I wouldn't say that way. But they didn't react that much to the forced swimming test when they were adults. So we had an opposite effect of dopaminergic imbalance in the early life and checking the mice behavior when they were adult. That was nice. We were very interested about that. But with mice, we're checking this specific developmental window. That's after birth. And we didn't want to check only that. We have a question of, but what about the other developmental windows? And when we think about mice is very difficult with that because they are, of course, they are in the womb. It's very hard to study early development or with the early beginning of development of the brain. So for that, we chose zebrafish. And zebrafish, it's a perfect model. It's a very good model to study this beginning of the brain development. So for that, as you guys know, it's very interesting to study zebrafish because it's fast external development, so it can use pharmacological tools in specific developmental windows. It's easy to manipulate and control, and it's optical clarity, so it's easy to see as well. So for that, we started to study the early development of the brain in zebrafish, especially because it's very similar to humans. Even at larval stages, we can see there's like specific neurons with the specific markers, so gene markers, so we can see in mammals and in zebrafish, so we can organize, now we're understanding more about the circuits of the brain in zebrafish, knowing that specific genes are expressed in dopaminergic neurons or GABAergic neurons, and in would say like a dorsolateral pallium or subpallium. So we're understanding even more of zebrafish, and we're getting there. It's like we understand that it's very similar to humans. I know sometimes it's very annoying to think of science only as something to be translational to disorders or human diseases. Since I also have a lot of interest in evolution as well, but we know where the funding comes, right? We have to show there's also an application for humans as well. I don't think that way. I'm not human-centric, but, well, that's a way to get some money for funding our lab and do science. Another thing that's very interesting is that zebrafish genome that has like a 70% orthology with human genes, but on the other hand, there's a lot of duplication, lots of duplication. So we always have to be aware of this thing. And the other thing that's challenging when we think of zebrafish to think as a translational model to study dopamine receptors or dopaminergic system is that there's a lot of duplication again, and so in zebrafish we have a lot of dopaminergic receptors. It's very interesting when my students ask me, but Bruno, humans are more complex, right? When you think of it, humans are way more complex than fish. I say, are you sure? Like you have to think of what is the parameter you're asking? Because if you're asking about the number of dopaminergic receptors, zebrafish is way more complex than humans. Humans have like 5 genes. Where's humans here? It's not even humans here. See, it's here. Yeah, 5 genes for dopaminergic receptors, and zebrafish has like 13, 14 genes for dopaminergic receptors. So regarding that specific parameter, zebrafish is way more complex than humans if you think of the number of genes of these variables. Well, okay, so we know the dopaminergic system in zebrafish. We are learning that. But what about dopaminergic intracellular pathways? Like I said, in some of disorders, we have alterations in DARP32, AKT, and CS1. So in a lot of dopaminergic intracellular pathways. But what about zebrafish? What do we know about zebrafish? We don't know a lot. So one of the things that we did in our lab is to sequence DARP32, to clone and sequence the gene of DARP32. So we checked the gene of DARP32 if it is in a chromosome 19. And after we sequenced the genome of zebrafish, we did like all the data analysis, the protein sequence and alignment, and we can see that zebrafish, it has the phosphorylation site turning 34, but in zebrafish it's 44. But it doesn't have the other phosphorylation sites. So the threonine 75, a lot of them we don't see it. I don't know. That's a question that we don't know the answer yet. If this was added during development or was deleted during evolution, or was deleted during evolution, we don't know yet. But that's one of the questions that we have. And also these other phosphorylation sites, they regulates the phosphorylation of threonine 34. So maybe zebrafish is more like it activates or it inactivates, and it doesn't have a more like a balance or like a very fine-tune of phosphorylation of DARP32. That's another question we don't know, but that's something that is in our mind. And then with our student, Beatriz Codo, I'm going to call her Bia. It's easier for us. It's a nickname. It's easier to talk about her. We checked what about the regulation of DARP32 in the zebrafish brain. So we did all the experiments now are in larval stages. Okay, it's between 3 days or 5 days old. So post-fertilization. So what we did, we checked the phosphorylation of DARP32, and we saw that DARP32. There's a decrease of phosphorylation of DARP32 when we expose the larvae, the 5 days old larvae, with dopamine for 5 minutes. With 10 minutes, we don't see that much. At 30 minutes, we don't see that as well. But at 5 minutes, at 100 micromolar, we can see a decrease of phosphorylation of the DARP32. And another thing that we checked is where DARP32 expressed. And we can see there's a lot of DARP32 genes. Here it was, in fact, it's confocal. So we can see the protein of DARP32 in the subpallium and in the pallium as well. And in the dorsal part of the pallium and subpallium as well. which is very similar to humans. So in mammals, we have the expression of this gene in the same regions as well as in zebrafish. So after that, we thought, like, what about our AKT? So to see if AKT is regulated by dopamine in the developing brain, we expose the larvae, the 5 days old larvae, with dopamine as well. And we can see here 100 micromolar worked really well. We can see here again. And the decrease of phosphorylation of AKT with dopamine happens after 30 minutes of exposure, not at 10 minutes, at 60 minutes we don't see as well. But when we expose with dopamine, we can see a decrease of phosphorylation of pallium, subpallium as well. And what is interesting when we check both AKT and DARP32 is that in mammals, Beaulieu, for example, showed that in mammals, the DARP32 is regulated in the first 10-15 minutes, and AKT is regulated around 60 and 90 minutes. Okay, zebrafish, we didn't see that. It was a little bit faster. It's 5 minutes, and here's around 30 minutes. But we can see there's also the same dynamics. So at 5 minutes, you have a decrease of phosphorylation of the DARP32, and of AKT is only around 30 minutes when DARP32 is not decreased anymore. The phosphorylation of DARP32 is not decreased anymore. So you can see a very similar dynamic of phosphorylation of DARP32 and AKT. DARP32 is a little bit faster, but we don't know yet if this difference in the dynamic of DARP32 and AKT phosphorylation, we don't know if it's because it's a larvae stage, or it's because it's zebrafish. So it's the difference between zebrafish and mice and mammals. Anyway, so now we know a little bit more about intracellular pathways regulated by dopamine in zebrafish larvae. What about the role in development? So for that, which we used, we wanted to aim, we wanted to choose one specific neuronal type. So not open too wide, the question shouldn't be too wide. So we checked GABAergic system. So for that, we use zebrafish, a transgenic zebrafish with GFP expressing in DLX6, which are GABAergic neurons. And we use the zebrafish and checked. We can see that a lot of GABAergic neurons in the pallium and subpallium and the ventral thalamus and the preoptic region. But especially here, I want to check here because that's where DARP32 is highly expressed in zebrafish as well. So using that specific zebrafish, the transgenic zebrafish, we expose the larvae from the 3rd to 5th day of hours post-fertilization. So it's for 2 days, and the 3rd day to the 5th day. That's a time window that's very important for the second wave of GABAergic system development. So it's a boom of GABAergic neurons migration and neurogenesis. So for that, we treated for 2 days. We exposed for 2 days with dopamine. We can see that there's a decrease of GABAergic neurons in the pallium subpallium of larvae exposed to dopamine. We can see here with MPTP. So like Ekker showed, MPTP is very toxic for dopaminergic neurons. So in this case, it would be last dopamine in the brain of larvae. So we can see there's a decrease of GABAergic neurons as well. But then we treated with MPTP plus dopamine. So we killed, this toxic. So we killed the dopaminergic neurons, but we added the exogenous dopamine in the embryo medium, and we rescued the number of GABAergic neurons. And again, with Kimperol, so dopaminergic agonist, we can see a decrease of GABAergic neurons in the Kimperol neurons. And here, when you check different regions, we can see that there's this inverted U curve. So less dopamine with MPTP, too much dopamine with the dopamine exposure, we have less GABAergic neurons. So when we think of the concentration curve, you have a inverted U curve in the number of GABAergic neurons. But the thalamus, we have a different response. It's like a linear increase of GABAergic neurons. In the preoptic, we have a plateau, and different regions like the pretectum, we don't see alterations at all in the number of GABAergic neurons. So what we saw again is an inverted U curve with MPTP and dopamine, and here the control levels. After that, we checked, let's see what is happening with the other cells. So we checked glia cells, and we didn't see any, at least rough alterations in glia cells when we exposed the larvae for 2 days with dopamine. So we don't see a rough alterations, and not like a strong alterations. And also we didn't see a strong alterations in number or morphology alterations in dopaminergic cells as well. So we can see specific to GABAergic, I wouldn't say it's specific, but we don't see alteration in dopaminergic cells. We don't see alteration in glial cells, but we see alterations in GABAergic cells when we expose the larvae to dopamine, to increase of dopamine during the specific time window. After that, we checked the percentage of behavior, so of motor behavior, so a number of initial movements, and to see if these alterations, the morphological alterations, would be also correlated to any behavior. So we check the motor behavior. We can see there's a decrease of motor behavior with MPTP treated, dopamine treated, and Kimperol treated larvae. But when we treated with MPTP plus dopamine, we have an increase of motor behavior, okay, using open field. But when we did light dark box, we saw a decrease of percentage going inside of the dark, of larvae moving to the dark side when they were treated with 5 days, 5 days from 3 to the 5 days post-fertilization. And after that, we thought, what about during development? Does it change and affects the motor behavior when they are older, a little bit older? So we checked the motor behavior at 7 and 14 days. What we could see is that there is still a decrease of motor behavior in open field test, even when they are 14s, but not in all parameters. In some parameters, they don't change, they rescue, or they come back through the ages, through the next few days. And to check the intracellular pathways, we use AKT inhibitor. What we saw that when we inhibit AKT pathway, we also have a decrease of GABAergic neurons. That was interesting. Again, dopamine, so the beginning of the signaling, and then inside of the signaling, in the bottom of the signaling, with AKT, we saw there's a decrease. So probably through AKT pathway, there's a decrease in dopamine regulates the GABAergic development. And then we thought about what about DARP32, but since there's very little, we know very little about DARP32 in zebrafish, we use morpholina to knock down the gene of DARP32, and after knocking down the gene, what we saw is that there is a decrease with 2 different morpholinos. There's a decrease in the number of GABAergic neurons, especially in subpilum, but not in the other regions. So DARP32, blocking DARP32 expression probably is also involved in the development of GABAergic neurons. And when we check the behavior, we could see again the decrease of motor behavior when we knock down DARP32 genes in the beginning of development. So with that, what we can see is that dopamine regulates the GABAergic system development, and that leads to alteration in of behavior emergence as well. And it seems to be that DARP32 and AKT intracellular pathways are involved in this role of dopamine in development of the brain. Well, that's it from that's what I wanted to say. Talk about my lab. Of course, I want to thank Professor Vincent Tropepo, University of Toronto, for the collaboration. Also, the Professor Marco Aurelio Romano Silva for collaboration from UFMG. All the funding agencies. We're struggling in Brazil. But with them, without them, we couldn't do anything, of course. So all the funding agencies from Canada and Brazil, and my whole lab, the whole group. Of course, you can see here this pre-pandemic. We can see all the smiles in the face, so it's pre-pandemic for sure. And if anyone wants to know more about the lab or to know what our research, everyone is invited to follow us. Not only the lab, but also my personal. Twitter or Instagram or Facebook, you guys you're all invited to follow us and to know more about the lab. Thank you again. It's very weird when you chair, right? It's not the nicest way, because when you say thank you, and then you switch the roles, and then okay, again I have to go back here. We switch the roles, and I'm back to the chair role now. So now again, the questions are going to be in the end, and for the next talk, I would like to call Professor Sue Gu for the next presentation. Sue, thank you again. Thank you very much for giving your time to talk about your interesting data. It's very interesting for me because of both Ekker and Gu, I follow them everywhere, and it's a very good pleasure. It's very nice to me to be here presenting you both, since I always cite you guys and I always follow you in your presentations everywhere in the world. Good, thank you very much. And again, the floor, not the floors, but the screen is yours.
Yeah, thank you so much, Bruno. Yeah, very, very kind words. I'll start sharing my screen. Let's hope it works. Entire screen.
I'll let you know.
Now. You can see my screen?
Yep, it's not full screen yet.
Okay, let me make it full screen. You can see my full screen?
Perfect.
Great. Yeah. So I really like to take today's opportunity to tell you about our journey in search of neuroprotective therapeutics and a zebrafish story. So my lab at UCSF is interested in understanding the inner works of the brain, and we use zebrafish as our model organism. As you can see, as all you know, zebrafish is great for genetics as well as cell biological studies, together with behavior as well as neurodevelopmental and neurocircuitry studies. So my lab interested in sort of three main areas. One relates to brain development, one relates to brain function, as well as brain maintenance. We also do a little bit modeling of disease mechanisms, but that's like a small effort in the lab. For today's talk, I like to focus on a story that we have recently developed and published on drug discovery for neurodegeneration. So here is the outline of my talk. So I will first tell you about how we have validated a chemogenetic dopamine neuron degeneration model in lava zebrafish as a good system for small molecule drug screening. And then I will tell you briefly about a small molecule screen that we have conducted, where we identified a G protein coupled receptor signaling pathway known as the Renin-angiotensin system signaling pathway. After that, I will tell you a little bit about the mechanism of how inhibiting the RAS signaling pathway can protect neurons. Finally, I would like to share with you that this inhibition of RAS signaling, being neuroprotective, is evolutionarily conserved across species. So this work has been recently published in eLife. So if any part of my talk is not clear or the some of the details is hard to see, I encourage you to check out this paper. So as you all know, Parkinson's disease, what happened? Can you still see the screen? Let me see. Okay. Parkinson's disease is the second most common neurodegenerative disease and the most common movement disorder. And the hallmark of Parkinson's disease is dopaminergic neuronal death. It is also remarkable to mention, I think as Marc has also alluded to in his talk, that the disease symptoms will not show up until more than 50 and 60% of dopamine neurons have been lost. As shown in this graph here, you can see the motor symptoms just starting to peak up right around when the 50-60% dopamine neurons are lost. And then when more neurons are lost, the symptoms getting even worse. So far, there is no treatment for Parkinson's disease except symptomatic management. So therefore, neuroprotective or disease-modifying therapies is in urgent need. So in contrast to the current conventional drug discovery pipeline that is used prevalently in the industry, we actually went back to a phenotype-based drug discovery pipeline. So this is actually a Renaissance approach. So before the current target-based drug discovery, drugs are usually discovered in a phenotype-based screen. So it usually starts with developing the assay and then perform the screen to identify hits and lead compounds, and then perform target deconvolution, and ultimately leads to target and understanding also the mechanisms of the hit and lead compounds. So for assay development, so we have been using this chemogenetic neurodegeneration assay Marc has also introduced to you. So this assay is basically utilizing this enzyme known as NTR, in short for nitroreductase. So it is a bacteria enzyme that has been used to ablate cells by specifically expressing this bacteria enzyme, for example, in dopaminergic neurons, as shown here. And then when a prodrug known as metronidazole, which is a very commonly used antibiotic in treating bacterial infections in humans. So when this prodrug is added to the water, for example, and then it serves as a substrate for NTR, and it leads to conversion of the prodrug into a toxic compound. As you can see, here is a around 4-day-old zebrafish, lava zebrafish with dopaminergic neurons in the control. But in MTZ-treated lava zebrafish, you can see there is profound loss of dopamine neurons. So however, we wondered what is the mechanism of cell death? Looking into the literature, there is not much information about how does the NTR-MTZ system kill cells. So because it's used to kill bacteria, so we wondered whether these systems also preferentially target, the genome of mitochondria, because mitochondria, has this evolutionary origin of bacteria. It is often said that a eukaryotic cell swallowed prokaryotic cell, right? That's how the mitochondria started in our cells. So to test this hypothesis, we checked for mitochondria as well as nuclear DNA integrity in control as well as MTZ-treated lava zebrafish. And then we limit the treatment to 8 hours is because we do not want to cause overt dopamine neuron loss, right, or fragmentation of the cell, which could leads to a secondary effect. So after treatment for about 8 hours with MTZ, dopamine neurons still remain intact. So after this treatment, we purify dopaminergic neurons and perform PCR using this primers that has been previously established to PCR mitochondria DNA. And it also yields a long product. And you can see that in the control, we can detect a pretty good DNA intensity, but in the treated samples, the mitochondrial DNA integrity is significantly reduced. At the same time window, it's interesting to note that the nuclear DNA integrity is actually not significantly different between control and MTZ treated, and likely because nuclear DNAs are protected by chromatin, in the nucleosomes. So we next want to know, what MTZ treatment does to the cellular dynamics of mitochondria. So here we used transgenic zebrafish that marks mitochondria in dopaminergic neurons. And here shows you the imaging of mitochondria in the control animal. So you can see the green dots are the mitochondria. And I'll play this video. You can see that in a control and healthy dopamine neuron axons, so these are the axons that project down posteriorly toward the hindbrain and spinal cord, as shown in this window here. You can see these mitochondria are moving around, and they're quite active. So like kind of like little cars on the highway. But when we looked at in the MTZ treated dopamine neurons, so here we treated the animals for about 16 hours, again before overt loss of any dopamine neurons or their terminals. But what you can see that there is significant reduction of mitochondria. And also the mitochondria does not seem to move much. And you can also see some rod-like mitochondria, suggesting that there are perhaps increased mitochondrial fusion. And we quantified these mitochondria dynamics by looking at the total mitochondrial number, the mitochondrial length, the mitochondria, how many mitochondria are moving around, as well as the velocity of mitochondria movement. And we observe all significant differences between control and MTZ treated dopamine neurons. And it's also interestingly, we also quantified the direction of mitochondria movement. In the control dopamine neurons, you can see that there's both anterograde as well as retrograde movement of mitochondria. But in the MTZ treated dopamine neurons, we exclusively only see retrograde retrograde mitochondrial movement, suggesting that these mitochondria are likely being targeted for mitophagy or for repair back to the cell body. So the question is, does this NTR-MTZ model have behavior defects such as locomotor deficit? So we looked at this by just recording zebrafish overall total distance traveled in the open tank. And then you can see that comparing to control, all the non-transgenic zebrafish treated with MTZ, the MTZ treated transgenic zebrafish show significant decrease in the distance traveled. We also performed this experiment in adult zebrafish. So for the adult experiment, we performed a chronic treatment with MTZ over like this 2-week period, and then monitor the total distance traveled. And we performed different concentrations of with the MTZ treatment, and you can see that there is a dose-dependent decrease in locomotor activity. We also tested whether this NTR-MTZ induced dopamine neuron degeneration can be rescued by Parkinson's disease-related genes activity. So what we did is to overexpress several genes that have been linked with human Parkinson's disease. So for example, Parkin, PINK1, and DJ1. So these 3 genes, when mutated, lead to recessive Parkinson's disease in humans. And when we overexpress these genes by delivering mRNA into the zebrafish embryos, what we observed is a significant effect in rescue dopamine neuron deficit, as for example, as shown here. When we treated with 9 millimolar MTZ, we observe a significant loss of dopamine neurons. And when we overexpress Parkin, we see a significant increase of dopaminergic neurons comparing to the control MTZ treated only. And the same has been observed for PINK1, DJ1 as well. And we also tested alpha-synuclein. Alpha-synuclein, as you know, is a gain-of-function mutation. So it, when mutated, it leads to a dominant form of Parkinson's disease. So when we overexpress alpha-synuclein, the mutant form of alpha-synuclein, we observed a worsening effect of the dopaminergic neurons. But the wild-type form, we did not see a significant difference. So to briefly summarize the part that I just talked to you, so basically we have established a dopaminergic neuronal loss model in lava zebrafish using the NTR-MTZ system. And we show that this particular system causes dopamine neuron loss as a result of mitochondrial damage. We show that the mitochondrial damage is not only at the level of mitochondrial DNA, but also at the level of mitochondrial dynamics. So this dopamine neuron loss phenotype can be rescued by overexpressing genes associated with recessive PD, and can also be aggravated by overexpressing gene, the alpha-synuclein, which is associated with the dominant form of PD. So next thing we did is to collaborate with a colleague at UCSF to establish a high throughput screening platform to look for small molecules that can potentially protect dopamine neurons from degeneration. So what we would, the assay that we would like to establish is a direct imaging of dopamine neurons. So I know that locomotor activity could potentially be used as an assay, but given there are many factors that can affect locomotor activity, we have decided to develop a imaging-based assay to directly look at dopaminergic neuron integrity. So using this assay, we have screened 1,400 bioactive small molecules and using quantifiable scores such as brand health score and the SSMD, which is basically correlated with the brand health score, but taking into consideration the variance across samples. And from this screen, we have particularly focused on a class of compound because three of the compounds that target different components of the signaling pathway have been pulled out from our screen, showing significant effects of dopaminergic neuron protection. So these are the names of the drugs, and these shows what this drug targets. So this is basically the pathway known as the Renin-angiotensin system pathway. So here is to show you in a secondary validation where we test more individuals using these compounds, and then we found that we can validate the screening results. So here shows you the image of dopamine neurons that are treated with DMSO or MTZ plus one of the drugs known as olmesartan. So the effect of dopaminergic neuronal rescue can also be observed at the functional level in terms of rescuing locomotor activity in lava zebrafish, shown in C, as well as in rescuing locomotor activity in adult zebrafish. So you might be wondering, what is the RAS signaling pathway? So the RAS signaling pathway is best known in the regulation of a blood pressure. So it's actually these inhibitors that we have pulled out from our screen are commonly used blood pressure medications. So this pathway starts with angiotensinogen, which is a pre-peptide that are secreted by liver. And then renin is a protease that's secreted by our kidney, will process these propeptide into angiotensin 1. And angiotensin 1 gets further processed by angiotensin converting enzyme into angiotensin 2. Angiotensin 2 is actually a very active peptide that can act on receptors, such as angiotensin receptor 1, to leads to blood pressure, leads to actually vasoconstriction of the blood vessels, as well as salt retention, and both of these will increase blood pressure. So therefore, inhibitors of this angiotensin receptor 1 can relieve high blood pressure and leads to a decrease of blood pressure. So in addition to this commonly known circulatory RAS, there is also a less known brain RAS pathway. So where the expression of these components have been detected in both neurons as well as glia cell types in the brain. So to further verify whether the chemical inhibitor's effect is indeed mediated by these signaling pathway, we use morpholino to knock down the angiotensin receptor 1. And here you can see knocking down both receptors. So there's 2 receptors in zebrafish as well as in rodents. So when we knock out both angiotensin 1A and 1B receptor, we see a neuroprotective effect that's comparable to treatment with the olmesartan, which is the NGTR1 chemical inhibitor. And here is just a Western blot, shows you that when we inject these double morpholinos, we could significantly knock down the protein, the receptor. So to briefly summarize this part of my talk, I told you about a bioactive small molecule screen where we have identified RAS inhibitors as dopaminergic neuron protective. So, and we also show that genetic disruption of AGTR1 activity can also be neuroprotective. So what is the mechanism of RAS inhibition in dopaminergic neuron protection? The question that we asked is whether inhibition of RAS signaling specifically in dopamine neurons will be neuroprotective. So this will allow us to determine whether it's actually the neuronal or the brain RAS that's playing a role, or whether it's the circulatory RAS. So to address this question or test this hypothesis, we used a conditional CRISPR approach. So what we did is we designed guide RNAs that can specifically target the AGTR1A as well as 1B receptor. And we co-inject these guide RNAs together with the Cas9 together with Cas9, in the UAS vector system. So we inject these into a transgenic zebrafish where the GAL4 is expressed in TH under the TH promoter. So therefore, it's expressed in dopaminergic neurons. So after that, we will treat zebrafish with either control or MTZ. And actually here we image the neurons before MTZ treatment as well as after MTZ treatment. The reason for doing that is because we want to analyze the same individual. And the experiment we conducted here is in the mosaic expressed transgene. And now we quantify this DA neuron intensity by basically using this equation as shown here. So what we have observed is that when we specifically knock down both AGTR1A and 1B using this CRISPR knockout approach, we can see a significant neuroprotective effect in dopaminergic neurons. So this result suggests that neuroprotective effect of RAS inhibition is dopamine neuron autonomous. Because the NTR-MTZ model is a chemogenetic synthetic model, it's not, Parkinson's disease model, sort of that shares etiology with human. So we have therefore tested whether inhibiting RAS signaling can also be neuroprotective in another model. So this particular model that we used is a Gaucher disease model. So as you probably know, Gaucher disease is a lysosome storage disorder. And due to mutations in this gene known as GBA, glucocerebral sidase. So individuals with heterozygous mutations in the GBA gene actually is one of the most prominent, populations to have Parkinson's disease. So what we did is use a chemical inhibitor of the GBA to create a Gaucher disease model in lava zebrafish. So what you can see here is first we looked at the behavior effect. So in the control lava zebrafish, if you put them in a like 6-well plate, they will swim around and you can see the tracks. But the ones that have been exposed to CBE, they move much less. And this locomotor deficit can be rescued by AGTR1 inhibitor, as well as by levodopa, because we know that levodopa can, supplement dopamine levels. So therefore, at behavior level, we can observe the rescuing effect of both olmesartan and levodopa. However, when we looked at the dopamine neuron integrity, what you can see here is that the levodopa is unable to rescue the dopamine neuron integrity, whereas olmesartan can. And here is the quantification. And also, interestingly, we see this rescue effects is somewhat specific to dopamine neurons, because when we quantify the serotonin neurons, we do not see an effect, because however, the CBE also affects serotonin neurons as well. So next thing, we want to understand the mechanism of how inhibiting RAS signaling can be neuroprotective. What we did is to carry out dopamine neuron-specific RNA-seq experiment. So here we used several conditions, including the control, the CBE, which is the GBA inhibitor, and then the CBE plus the olmesartan, which is AGTR1 inhibitor. And we also used the MTZ model and MTZ plus the olmesartan, as well as olmesartan alone. So here it just shows you the data. Because of the time, I'm not going to go into the details about the findings, but to point out that what's really striking that we found from the RNA-seq experiment is that the genes that are altered in common, shared between both the CBE, the Gaussian model, as well as NTR-MTZ model, are really mitochondrial genes. Further validate that the NTR-MTZ model really, is caused by mitochondrial damage. So we see genes involved, for example, in oxidative phosphorylation, respiratory electron transport, and so on, involved in this mitochondrial function. And remarkably, when both of these models were treated with olmesartan, the AGTR1 inhibitor, we can see a significant restoration of these mitochondrial pathway genes, the respiratory electron transport, oxidative phosphorylation, and so on. So to summarize this part of my talk, I've shown you that inhibiting RAS signaling is neuroprotective cell autonomously. So therefore, RAS signaling activation can act cell autonomously to promote cell death. And what RAS signaling, when it's activated, right? For example, the AGTR1 receptor, when it's activated, it somehow downregulate mitochondrial pathway genes. So therefore, when we inhibit this receptor activity, we can see a significant neuroprotective effect. So I showed you, the data to support this conclusion, including the cell type specific CRISPR knockout, as well as cell type specific RNA-seq experiment. So the final question I like to discuss with you is, are RAS inhibitors neuroprotective across species? Is it only neuroprotective in zebrafish? So to address this question, we first used an invertebrate model. So in Drosophila, actually, there's remarkably good models for Parkinson's disease. So for example, when in fly, a fly mutant in PINK1 will have muscle mitochondria defects, like mitophagy-related defects, and very obvious phenotypes that associated with muscle deficit. They also have a significant loss of dopamine neurons, which has not been able to be observed in rodent models, for example. So one of the remarkable and easy to observe phenotype is this wing phenotype. So you can see that in wild type Drosophila, their wings are, very nicely naturally positioned. But in the mutant of the PINK1, you can see their wings are held up. And when we treat the PINK1 mutant Drosophila with olmesartan, the AGTR1 inhibitor, we see a significant decrease of this held up wing phenotype. So another phenotype is this thoracic indentation, which is basically their thorax have like this indent in it, and this phenotype is also significantly decreased upon olmesartan treatment. So the dopaminergic neuron deficit has also been partially rescued in terms of both dopamine neuron number as well as the mitochondrial appearance. You can see that in the PINK1 mutant, the mitochondria really forms large aggregates in dopamine neurons, and then this phenotype has been completely alleviated upon olmesartan treatment. So the ultimate thing we want to know is the RAS inhibitors actually useful for in human Parkinson's disease patients? So we are able to address this question because the RAS inhibitors have been widely used as a blood pressure regulating medications, and including in PD patients. So by utilizing this data that is available from the Parkinson's Progression Markers Initiative, PPMI database, so this database has been put together through the Michael J. Fox Foundation. So we looked at their data, and this set of data is extremely clean because these patients undergo extensive clinical examination every 3 months. They also have very detailed records on what type of medications these patients are on. So including, for example, the blood pressure medications, what type of blood pressure medications patients are taking and how long and how often they take it. So there's very detailed records. So we started with 423 de novo patients, and then we basically, grouped them and then to look through the data to make sure that these patients really are qualified in our data analysis. So in the end, we identified 96 patients who are on the RAS inhibitors, as well as 212 patients who are not on RAS inhibitors. So among these 212 patients not on RAS inhibitors, some have hypertension. For example, 42 of them have hypertension, but they are on other type of antihypertensives, but not RAS inhibitors. So we look at this data in these different groups. And then what we found, the readout that we looked at is the time to levodopa therapy. So levodopa is still the gold standard for treating Parkinson's disease. But however, levodopa has this known side effects is that, after 5 years also, the drug effect starting to wear off. And also it can induce unwanted side effects such as dyskinesia. So because of that, neurologists tend to withhold prescription of levodopa until absolutely necessary, to the patients. So therefore, the time to levodopa therapy can be used as a way to quantify the disease progression. So you can see that in the RAS patients, the RAS PD patients, their average time to levodopa is about 32 months, whereas the people who are not on RAS inhibitors is about 25 months. So there is a significant delay in the need for taking levodopa, when the patient is on RAS inhibitors. So we also looked at UPDRS scores. These are just standard scores that neurologists use to quantify disease progression. We observed that UPDRS Part 1 score, which is to measure mentation, behavior, as well as mood, is significantly improved in PD patients on RAS inhibitors. However, the Part 2 and Part 3 scores, we did not detect significant difference. So to summarize the talk that I just shared with you today, so I told you about how powerful zebrafish is, that allow us to be able to carry out an in vivo high content imaging based small molecule screen that allow us to identify the RAS inhibitors to be neuroprotective. And I also present to you that these RAS inhibitors, especially, the receptors, the AGTR1, is likely to play an important role in dopaminergic neurons. So when there is neurotoxic insult, somehow the AGTR1 receptors can be activated, and then indeed leads to damage of mitochondrial function. So while we apply RAS inhibition, right, by these chemical inhibitors, we're able to counteract this effect of neurotoxic insult. This neurotoxic insult can be environmental toxins, can also be, genetic, due to internal genetic defects. So there are still, several questions we're very interested in addressing. One is, how is the pathway activated upon toxic insult? How does the RAS pathway get activated? We don't quite know. We don't quite understand how that it works inside the brain. And what are the downstream effectors of RAS? And obviously, the ultimate question that are more sort of in terms of therapeutics, these drugs that I show you, these RAS inhibitors, actually, they're not very easily crossing the blood-brain barrier. So they probably have very minimal ability to cross the blood-brain barrier. So that could potentially, explain why we did not see the UPDRS score 2 and 3 to significantly improve. So we're currently, working on strategies to increase this inhibitor's ability to cross the blood-brain barrier. So this work is really spearheaded by Jeffrey Kim, a recent graduate from the UCSF graduate program, and as well as by another graduate student, Harrison Liu is the Bio-E program graduates, and also Han Mo, which is a exchange graduate student from Tsinghua University in China. So this work is supported by National Institute of Health as well as Department of Defense, and with many of the UCSF collaborators as well as outside UCSF that have contributed significantly to this work. I'd like to thank you for your attention, and I'm very happy to answer questions that you have. All right, let me see, what do I do now?
Thank you, Sue.
Okay, do I stop?
Yeah.
Present? Okay, great.
Thank you. Mark?
I didn't take too much time.
No, it's fine. No, I in fact, I'm gonna confess with you. For me, this is amazing time. Talking about science, for me, it's way better than anything that's administrative, right? That's the thing that we have to put a lot of effort and time on that, and I don't think anyone likes it, right? I think we love talking about science. That's the best part. Anyway, so thank you for both of you, Sue and Mark, for your talk. I have some questions in my mind, but I'm gonna give space to the guys on YouTube. And one question that we have is for Mark. Do you guys have any hypothesis of how these neurons regenerate in 1 to 2 days in case of the larvae?
Yeah, I think the word regenerate here is probably, there is recovery for sure. Whether there's regeneration, I'm not so sure, because perhaps the fact that the larvae are making such large amounts of neurons, and because we've kind of stopped the treatment a couple days earlier, that might just be enough. So that's another reason why perhaps if the rate of production is lower in the adults or older animals, it might be useful to look at regeneration mechanisms at that stage.
That's good. I have another question, but before asking the question, Sue, do you know Robson Santos?
Whom?
Robson Santos. I'm trying to say in English. Robson Santos.
Oh, okay. I don't recall. Yeah, I don't know.
He's the one who discovered the receptor for angiotensin 1 to 7, and he's the one who described the alamandine. He's a colleague here in the department.
Oh wow!
Yeah, I know, but I'm not as good as him. He's amazing. He's emeritus. He's a very, very good one. And then when you presenting that, I was like, whoa! I think Sue maybe she would like to know that this is this is something that is big in the department. Everyone here, I don't do it, but a lot of people here studies angiotensin 1 to 7.
Yeah, that's amazing. So yeah, the angiotensin 1 to 7, which I didn't really get into, it's sort of the good ligand. Angiotensin 2 is sort of the bad ligand. Yeah,
yeah, that's true.
Yeah.
And now he's studying alamandine.
Yeah, I think it's ACE2 that processed it into the angiotensin 2 into the angiotensin 1 to 7, right? ACE2, as we all know, is the COVID receptor as well. It's the SARS-CoV-2 receptor. Yeah.
Yeah. And when you were talking, I thought like, oh wait, I have to see his paper, and then I just to remind of some stuff that he's studying, and then when I went to his Google Scholar. There's one paper that is cited now for like over almost 100,500 times, and it's mostly because of the COVID. Now he's he's doubled like the number of citations, and I thought, oh, wow, well, good luck for him for pandemic, right? Not for us. Okay, another question here is let me see. Wait, 1 second. Message retracted. So the next one. Congratulations to all the researchers for nice presentations. And I have a question for Dr. Ekker again. After exposure to the toxin, did you observe or evaluate any changes related to epilepsy in these animals?
No, we did not. No, but that could potentially be something very interesting to look into. No, we did not.
No. So no. The other one is Angelo Piatto. We work with zebrafish models of epilepsy, and we are interested in developing a model of damage followed by epileptogenesis, and toxin use could be a possibility. Yeah, but you said you didn't, right? So the next one is Anna. Anna asked, I have a question for Professor Bruno. Great talk, thank you. When dopamine binds to D2 receptor, there is decreased AKT phosphorylation, but how is this related to DARP32? Well, in fact, in this one, we don't know that much if there's any correlation, any interaction between both of them. There's some direct interaction, right? And there's some data suggesting that, but it's not like it's not a paradigm that's that I think it's like well done yet. So I don't think it's well supported yet. I still have some stuff, but in this case, it's because when D2 receptor is activated after a long time binding the dopamine binding to this receptor, D2 receptor, there's one protein called beta-arrestin. So the arrestin binds to dopamine receptor when it's phosphorylated, and it pulls the receptor to inside of the neuron, and then when this beta-arrestin binds to the receptor, dopaminergic receptor D2, and it puts it inside and pulls inside the receptor, then it decreased together with the PP2A. It decreased the phosphorylation of AKT. So first, through G protein, it activates the PKA pathway, and then DARP32, and then after that, the beta-arrestin binds the receptor, pulls it down to inside of the neuron, and then you have the decrease of phosphorylation of AKT because of a complex at PP2A. Let me see if there's any other question. And no, Angelo just said like thank you. I know you're very short because there's another table waiting for us, but I have one more question. Since Marc just answered the questions, now I'm going to ask Sue. Sue, regarding that, like the mitochondria, I was wondering, like I don't know, like I'm trying to when I was showing, and I love the videos of them like moving around, like the projections. And when you said like it's like a highway, I was like, no way, like I never seen that. I really liked it. Very but nice. Do you think it's something related, like it's direct signaling to the mitochondria, Or is it like you change the cell homeostasis and then the mitochondria reacts to that? Because when I think of mitochondria, I always think of people inside of a bus, you know? But the bus dies without them, right? I know it's a bad analogy, but I always think of people like a cell, like living cells or a living organism inside of a cell. And I wonder if it's like, I always think of a homeostasis and how the mitochondria act or behave, changing homeostasis of the cells. So do you have any hypothesis for that? Or like, I don't know.
Yes. So are you asking about in the model or whether it's the RAS pathway?
Both.
Or maybe both.
Yeah.
Okay. Yeah. So with the NTR-MTZ model, right? So the the metronidazole, right, is an antibiotic.
Yeah.
Right? So I mean there is some there's one paper that I was able to find shows that metronidazole, the mechanism of metronidazole as an antibiotic, like how does it work, right?
Yeah.
So there's some chemistry involved. I don't remember the details, but it's basically it can attack DNA. It's shown that this chemical, right, metronidazole, once it's converted by the NTR, by the enzyme, right, which only expresses in bacteria, by the way. So it's not expressed in us unless we engineer it, right? So that's why when we take metronidazole, we don't get hurt, but only the bacteria gets killed, right? Mm-hmm. The reason is because bacteria express nitroreductase, and that converts the metronidazole, a prodrug, into a cytotoxic compound. And the cytotoxic compound works by attacking their DNA. So there is some evidence about that, okay? Mm-hmm. So suggesting it's a very direct effect, attacking DNA, right? Like cause DNA damage. So the question is, does that also happen when we express these NTR in dopamine neurons, right? So now the dopamine neuron is kind of turning into a bacteria, right? Because it's now expressing NTR just like a bacteria. So now they can convert MTZ, the prodrug, into the toxic compound, right? Yeah. So the question is, what does this toxic compound do inside dopamine neuron, which is now like a bacteria, right? Expressing NTR. So we hypothesize that it probably could do the similar things as it does in bacteria, which is to attack DNA, right? So therefore we directly looked at the DNA integrity. So we looked at both mitochondrial DNA and the nuclear DNA, right? Mm-hmm. And obviously we don't want to, have this, when cells starting to really fall apart, right? Like dying, everything fall apart, as you were saying. The whole thing, is like a catastrophe, right? Everything's breakdown, right? So therefore, we looked at the DNA integrity at earlier stages upon MTZ treatment. It's like 8 hours after we add MTZ, when the cells still look completely intact. That's how we can purify these cells, right? So we purify these dopamine neurons, which is now like a bacteria, and then specifically extract the total DNA, and then we PCR with the mitochondria-specific primers, mitochondrial DNA specific prime and nuclear specific DNA prime. That's what I showed, that the mitochondrial DNA is really, really, breaking down because we couldn't get, a good band, the product, right? Whereas nuclear DNA is pretty much intact at this stage because, they're protected. Nuclear DNA is inside the nucleus and protected by chromatin, right? By all these nucleosomes, lots of proteins. So that's how we think that the onset of this dopamine neuron loss is due to mitochondrial DNA damage. So now the question is how RAS inhibitors, right? How the RAS signaling pathway gets involved. Is this a direct effect or is it somehow indirect effect, right? That leads to the protective effect, right? So that part we still don't quite understand yet because this receptor, this AGTR1 receptor, has been shown to be expressed in many places inside the cell. So not only on membrane, because we usually think, a G protein coupled receptor expressed at the cellular membrane, the plasma membrane, right? But there are papers published to show that these receptor can be expressed on mitochondria as well, but can also express on the nucleus. There's papers, showing that these G protein coupled receptors are localized in many different subcompartments inside the cell. So it could be a very direct effect. So the question is how these, in your toxic insult, right? Like the NTR-MTZ, even the CBE, which inhibits the GBA, how these leads to activating these signaling pathway, the RAS signaling pathway. That part we don't quite know. We don't know how it that's why I think that's a really exciting question, is to understand, is it upregulated ligand, for example, the angiotensin 2, or is it some other mechanism that causes this receptor to be activated, right? Once they're activated, it can do bad things to the cell. Yeah.
That's cool.
So that's basically our current understanding is.
That's really cool. Yeah, Mark. Yeah.
So, the NTR NTZ system has been used to ablate cells in many other systems in zebrafish. So do you hypothesize that similar mechanisms are? I really like the mitochondria DNA experiment, but and presumably that's going to happen in other cell types. But what happens downwards? What would you think happened downwards in these other systems? For example, some of the first NTR, MTZ papers in zebrafish were used to ablate cells in the pancreas.
Yeah, that's a really, really good question. So yeah, definitely this is a very universal system. You can ablate any cell types that you're interested in, right? So I would hypothesize that this is a common way of how the NTR-MTZ system works, is to attack mitochondrial DNA. But what happens downstream maybe could be cell type specific, right? When mitochondrial DNA is damaged, right, in dopamine neurons, for example, it could trigger a cascade of mitophagy, and things like that, right? In other cell types, it could be slightly different things gets activated, right? And different cells have the different detoxifying mechanisms, right, in response to mitochondrial DNA damage. But so far, I think if we give enough MTZ, like all cells seems to die, right? So it's a very robust system. So I think this mitochondrial DNA damage is bad for all the cells. Yeah. So it's a very powerful system to kill cells for sure.
Well, we're gonna finish now because there's another table after us. I said that I wanted to talk to you guys after, but unfortunately I have to go to the other table. I tried to skip, but I can't skip. But anyway, I would love to talk to you guys again, maybe in the near future. I can contact you, both of you.
Sure.
Thank you very, very, very much for you guys to giving again your time. You can see they're installing an air conditioning here. At least it's an air conditioning. It's pretty good. But thank you very much. They're asking one more question. Yeah. Okay. No, they're gonna kill me. But anyway, I'm like I said, as Steven Spielberg here, so I can control. So there's another question. Anna asked another question now for Sue as well. Does dopaminylation play a role in your model? That was interesting.
Does dopamine ablation?
Dopaminylation. Dopaminylation. I was saying it wrong. Dopaminylation.
Dopaminylation. What does it mean?
It's something I don't remember that much, but it's something like it is toxic. I'm not going to be the good one to explain that.
Oh, so whether the dopamine itself is like the dopamine toxicity contributes to this?
Yeah.
No, that's a really interesting question. So, I mean, there's all these hypotheses about, right, why Parkinson's disease, right? Why dopamine neurons selectively die in Parkinson's disease, right? Given that a lot of these toxins, or especially those genes involved in PD, are actually expressed in almost all the cell types, right? In our body. So there's this dopamine hypothesis, is that because dopamine, it's very toxic, right? It somehow increases the vulnerability of these cells. I don't know if it plays a role in our model. So one way to test that would be to silence the TH gene, right? So like to get rid of the endogenous TH gene, and then see if the neurons don't die as much. Yeah, but that's an interesting thought. It's definitely, there's this dopamine hypothesis of Parkinson's disease.
And I found something that was interesting. I think I'm saying it wrong. It's a histone H3 glutamine 5 dopamylation. So it's a post-translational modification. I didn't know that.
Okay.
Yeah. Do anything about that?
Histone, right? No, I don't know. I don't know if it will. Yeah.
Me neither. Yeah, I'm sorry. Yeah, but I also don't know. Well, again, so thank you very, very much for your kind and for being here with us. I hope in a post-pandemic world we can meet personally and maybe the dopamine, right? Is it next year? Yeah, next year, right? The dopamine in Montreal, the conference. I think it is. We'll talk to. I'll talk to you guys later. I think it is. But thank you very much again. Do you guys have any last words for the Brazilian people here that are watching us?
Thank you.
I want to say thank you for having me. Yeah, it's really fun to discuss science. Yeah.
Yeah. Thank you very much. Okay, see you next year,
Yeah.
hopefully.
Bye-bye.
Bye.
Bye, Bruno.